right, welcome everyone to today's SKC Science and Technology webinar series. I am Dept, organizer of this webinar series, and it's a pleasure to have today Professor Upendra Nongthomba. So he is a very well-known scholar, a professor at the Indian Institute of Science, as you can see it from the first slide that he has posted here. He is originally from Imphal, Manipur, and he then studied in Delhi, I think, in school also, then in Mysore for his undergraduate, master's and PhD. And then he went to York University for postdoc, and he was back from there in several capacities, and then joined IISc, I think around 2006 or 7. So it's my...
No, I joined in 2004.
2004.
It has been, yeah, long time.
So I was off on a couple of years.
No, that's fine.
Yeah. So it's my great pleasure to have you today. And I actually found out about Professor Nongthomba from one of my friends, actually. And I thought I will have him as one of the speakers on these things. And it's always a pride to have somebody from Northeast doing so well, in the rest of the country. So with that, I'm going to hand it off to Professor Nongthomba. So the way we do it, as I say, it is more like almost like a discussion fireside chat. So once in a while, I'll stop and ask you a couple of questions to clarify, okay, what do you mean? As you probably guess, I am not also in this field, but it's a very intriguing topic, to talk about. So with that, I'm going to give it to the professor. You can go ahead and get started.
Professor Deep, thank you very much. First of all, thank you very much for giving me this opportunity. It means it got postponed many times because of the health condition of my mother. She is okay now. So it is a pleasure to interact with all of you. Today what I will do is that you said that I should have 15 slides, but today what I will do is I have got more, but those are data which can be skipped. But the whole essence is in the first 15, 16 slide, that why we do such research and why model organisms are required. And if anyone wants to clear things, they can stop me anytime during the talk and they can clear it. And I will try to, convince you that what we are doing, why it is so important, and why we get excited. Because at the end, you can do science. Professor Deep must be knowing it, or many of you, those who are senior here, attending will know it. One is that you do it because you are supposed to do it to get a salary and take care of your family, or another is that you do it because you love doing it. And at the end, personally, my background is from politics, but I am the only one in science from my family, and I'm the only one from Northeast in the Indian Institute of Science, and I have to make sure that the
Looks like we lost him.
message. And the second thing is that I'm into science because every day when I go to lab, I get excited, right? I feel that we are going to solve a
looks like we lost him. Professor Upendra, we lost you for a minute. So if you can repeat what you said. Okay.
Yeah. Can you now see me?
Yeah. Now I can see you, but you have to reshare the slide, I think. So.
Yeah. Okay. Just 1 minute. what happening? I don't know. Somebody threw me out of this thing. I don't know that. Can you see my slides now?
Yeah, and if you can go to the full screen mode.
Right. Okay. So at the end, however, when we at the end of the day, we may not get any breakthrough, but disorder condition and try to find a cure or the precautions, right? If we can generate some idea, that is what we are looking at. So I will just share why we do it. Then at the end, I'll give you few examples, and you don't have to see the whole data, even though I have the data. But if you want to, then you can just go through all my papers, and somebody can write back to me if you have any problems, right? Okay. So we all know about the genome. Genome is the whole genetic constitution of every organism, and of course we are much more interested in the genotype phenotype correlations of if there is a defect in the, so whatever we are, it is because of the our genotype. And if and whatever phenotype, whatever we exist as a external thing, is because of the our genetic system, right? And it varies from individual to individual. And today we know that even and the whatever 120 more trillion cells in the our body, adult body, even they can vary because the moment the cell divide, they are heterogeneous, right? And so disease can happen. Disease is nothing from the genetic point, any abnormality. Sometimes abnormality is good because any difference, what we call as the descent with modification, that is the precursor to the evolution. And so we are today because of that changes which happen over the generations, over years, billions of years, maybe that is what people claim today. But most importantly, sometimes what happens is the change in our genotype can make us hamper our normal growth or development. Sometimes we can be very susceptible to the disease conditions, infectious disease like say bacteria, virus, and things like that, right? So we have close to like 24,000 genes, but if you look at it, every even if you count out of say supposed to be 3 billion letters, right, for the every body, I mean, you know, we will have highly conserved sequences, but every genome, even if you consider 2,000 minimum, let's say 2,000. Whereas the dystrophy, which is the longest, I mean, gene, it can go up to 1.78 million. But let's say generally, if you take only say 2,000 base pair for the every gene, you are talking about say 2,000 into 24,000, right? Okay, that many mutation can exist. I'm just talking about the coding sequence. Now you include that with non-coding, then regulators and things. So many phenotype can exist.
So a very high level question, given that with the base pair, the billions we get to, on an organism, typically what percentage is changes with a mutation? It's very, very small still, isn't it?
Yes, Professor Deep, but today, in terms of the coding for the genetic coding, we are talking about those which will produce DNA or the important RNAs like say ribosomal RNA or some others ribosomal RNA. But today we know that even what we told as the junk DNA or any which are not supposed to be coding, today we know that actually they can generate something called as the long non-coding RNA or say small non-coding RNA or some kind of thing. In this RNA itself, actually, the way they organize it actually can influence the expression of a gene. So I think though we say that, only a small part of the DNA or the genome or RNA in the virus will code it, but in reality, we are talking about that every part of the whole genome will play one role or another for the expression of the gene, something which we don't know yet, right? Except for the few expression of the few genes or the say chromatin organization or those who study extensively, or otherwise we have, I think we know nothing about actually what happened at the genomic level. I mean,
Okay, got it. Okay.
yeah.
Mm-hmm.
So this is what actually excited us to look at this genotype phenotype correlations and the differences what we expect at the genetic level, if what kind of disease they can cause. If the whole idea is that many people say that, Upendra, the infections happen and there are cardiomyopathy which has been happening, and you don't need to know the genomic level. I mean, if there is a brachycardia, I mean, tomorrow you put the pacemaker and you don't need to know the genomic region. I mean, right?
Mm-hmm.
This has been happening for years, right?
Right.
And I agree that there are many conditions where we know the phenotype and we treat the phenotype. That is the way our medical science has been going on, have been evolved. But the whole thing is that there is a catch again. If we knew personally that if you are going to be when you reach around say 50 or 60, you are predisposed, we call it predisposed with the say brachycardia, or you are predisposed with the diabetes or something, wouldn't you take some precaution, right? That's the whole thing. I mean, and that is what we are looking at. So the we means that those who work in the model organism, that is the whole point, that we want to make model for each one of us. 6.5 billion people, if possible, 6.5 billion model organism. We call it as the model avatar for each one of us. And we should be able to say, instead of you eating any kind of food, we should play around maybe with this avatar and say that if you eat this kind of diet, then
Mm-hmm.
you will be prone to developing obesity, right? Okay, or that kind of thing. That is what, or say diabetes or the cardiomyopathy. So you can live your life through the
Mm-hmm.
avatars of the
Mm-hmm.
model organism, and that is the whole take. My second takeaway from this talk is that the model organism research actually can influence all these things. And the third thing is that the moment you have the disease conditions, as I told you, even when we have such a conserved sequence, right, at DNA level, right, because of that, we are so similar in terms of our phenotype, but each one of us are different, right? And when we are different, the drug, whatever we develop, also can be different, and that is where we call as the personalized medicine or the precision medicine comes. And that is what the model organism again can help, is that whether a medicine work efficiently for those people, that is what we can look at, right?
So
So
here,
that,
if I may interrupt you for a second,
yeah, please,
Professor.
please, please, please.
So the first question is about the model organism. The
Yeah,
question is more general. How do you know a particular organism is actually a model organism? Is there some
yeah,
sort of a benchmark or some kind of a markers that based on that you can say, okay, these model organisms are model organisms for the purpose of understanding
yeah.
human things, right? I mean, we
Just
talk
give me,
about lab
yeah.
science. Okay, you do the mice, that has been done forever, but
Yeah, yeah,
that's kind of a
absolutely.
yeah.
You catch hold of that thing, and I will discuss you in 2, 3 slides more. Then I will
Okay,
just
that's
come
fine.
back
That's
to
fine.
the model organism. Yeah.
Great, thank you.
Yeah.
Okay.
Okay. So ultimately, for the every individual, is that if you know this genotype for phenotype correlation, ultimately you want to discover a drug which should cure the things, right? And for developing such drug or the interventions, therapeutic interventions, you need to know exactly, I mean, what is the gene what is being disrupted at the what stage and things. For example, say I'm just giving you the recent thing, which is the, like, if you give at the right time this 16 crore injection, and it has to be given before the 18 months of the baby is born, without what we call as the SMN protein, right? Spinal muscular dystrophy, that uh, protein is lacking, then you give it from outside, and actually it can rescue the individual. All right. And all of us, those who work in genetic field, this is exactly, this came out of the basic research first, right? Okay. And all the details of the gene was extensively studied. Then some pharmaceutical company took it over, and they made the drug, right? Okay.
Right.
But
Mhm.
what about the small thing the basic research can provide? I mean, what time the gene get expressed, what is its role, and what time if we give say a normal sequence, because here the protein is not being produced. If we give it from outside, how long it takes to get, how much we have to give it tissue specific way, because it should not have any side effect and things like that. So there are many way drug kinetics, which let's not talk about those things, that is pharmaceutical, that will go to the next other thing next day, maybe next time when I talk about it I can discuss in detail. But today, the what those who are working in the genetics field, what we really want is that we want to develop drugs or something for every disorder in the world. I mean,
Mhm.
as I
Mhm.
said it, right? Okay. So if you understand it, then you don't need to give a very expensive one, because there are 4 different kinds of SMA protein mutations, where the mildest version can survive. The people, those who are means have a point mutation, some of them survive till the adult stage, but they are handicapped. Such people can take no, motor neuron survival drug oral, which can be taken orally. For example, here every SDI is oral drug which can be taken. This allows the motor neuron to survive for long for this SMA patients, right? Okay. So this is the crux. I mean, I'm just giving you example. I mean, this is so expensive, but this is where we want to make a cheaper version also. Uh, and because for the pharmaceutical company, it is about the business. But if we can make it alternative versions cheaper, that is
Right.
also a challenging thing that we want to look at in future, right?
Mhm.
So
Of
these are
course.
some of the human genetic order where there is a genotype phenotype correlation is quite well known. I mean, in other words, if there is a mutation in this part of the gene, you will suffer from this phenotype, right? Okay, this is quite well known. And why we should worry about the genotype phenotype correlation is that at least now today, as of today, I mean, there are 3,645 genes known so far, where genotype phenotype correlation is quite well known, right? These are some of them are here. And once you know the genotype phenotype correlation, at least some of these are, like, the individual child will not survive, even if they are congenital, we call it, right? They will not survive more than, say, 2 years, even if they are born. So if you have these uh, high genotype phenotype correlation, if you know it, the idea is that at least you can take a prenatal screening test, and you can um, take the what we call as the informed decision whether you want to continue with the pregnancy or not, and all these things. I mean, it comes into eugenics, which again is a what we call a big field to discuss, and I'm not here to discuss that.
Mhm.
But what I'm trying to say is that at least we should know that if you have a likely disease, whether you want your children to have that disease or not,
Right.
and
Right.
whether if they are going to have that disease, whether what are the precautions they can take. That is what the whole genetic disease screening is about. And today, as of today, we have
close to 2,645 genes, which have been extensively worked out genotype phenotype correlations. And at least in India, there are companies where you pay 18, 19,000, and those company can provide you the details with a small blood from the neonatal baby. I mean,
I see. Very interesting.
yeah,
Okay.
right.
Mhm.
So this is
So
I
if you
told
could go back to previous slide, I have one
yeah,
question about
yeah,
the gene. You listed this, you know,
few
FMR.
of them.
Yeah,
Yeah.
I understand, but
Yeah.
FMR1, FMR2, is there an international standard on how they are named? Just out of curiosity, like when you say,
Um,
or is it just kind of a anecdotal way of naming those genes?
People used to name according to the what it started with the Drosophila people. Drosophila
Uh,
people naming all the genes,
mhm.
like say spaghetti if it is looking
Okay.
like this, or the wingless if they don't have the wing and phenotype, right? Okay.
Uh-huh.
So some of the gene names are derived from the Drosophila thing because they are very well-known mutation in the Drosophila, so they retain it. But now today, as of today, we have the OMIM thing where the International Genetic Society, they name these protein names. I mean, so the protein names associated then the what we call as the disease condition. So for example, say DMD or the Fragile X. These are DMD. We used to dystrophy, Duchenne's muscular dystrophy. Today, the patients are called as the dystrophic, and the gene name is DMD. Whereas it was known before 1990s and 2000, it was known as the dystrophin.
I
But
see.
today we
Okay.
know it as the
Okay.
DMD. So it keeps on changing.
I
But
see.
most of
Mhm.
these names are now internationally known as of in OMIM database, which is the PubMed International
Okay.
Genetic Society accepted norms. I mean,
Okay. Okay. Thank you.
yeah. So this is very well known. We know that hardly around 25, 24,000 now, actually. This is very old slide when the genome came out, but protein can vary, and there are some different splicing. Genes can undergo what we call as the different forms, I mean, from the sequence itself. Then those splice variant can be modified by what we call as the post-translational modification. This is what we call as the crux of central dogma, and this is what we think that we know it. And today, however, the question comes is that what stage the gene is required, right? That is the functional requirement, because if you really want to know a disease or the curing thing, you need to know when a gene is required functionally, right? When it is expressed and when it is required. That is what I'm just giving you one example from Drosophila, but this applies to all the genes including human. Okay, and I have put a Drosophila data because we have like embryo larval in the same way, like you could have prenatal, then the baby and the things like that, you can have it. And this is something which is challenging, but however, the 3 labs in the world have been given money by now Allen Institute and Google to generate database for the all the developmental stages, gene expression pattern, then the organ, particular specific organs. So they have been given huge money to generate all this database. Maybe in the next few years, we will have the whole database. Already brains, at least the genes expression pattern of the adult brain, it is already available in the Allen's Institute if you look at. Similarly, we want to do it developmental stages or the tissue specific or the organ specific expression of the genes. And this is important because you want to target a gene or the protein in a tissue specific way, and so that the curing, if you are looking at, it should be tissue specific. That is what people are looking at, right? Okay, so these are all expression level. Let's not worry about it. Let's don't look at the data. Just take it from me that we need to know the requirement of the each protein, how much fold is required. And some of you are filling too much data. Please don't sleep. I will skip some of this data, right? Okay, because I just want to let you know that today, even though we talk about the 25,000 genes, but today when we look at the physiology of the each organ, even though a gene can be expressed in an organ specific, say heart, but sometimes what people have found is that actually that protein can cross-talk with the other organs, right? So what we call as the organ-organ communications, right? Okay, which was not there before. We knew that there are endocrine system, exocrine system, all these things are there. But today we know that there are exosomes. I mean, I'm taking all these names, but I'm not trying to convince you. What I'm trying to say is that there are so many things which were not known before, but today we know that there are circular RNA, circular DNA, which can go from one tissue and can reach other tissue, and they can influence the expression of the other genes, right? So organ-organ crosstalk is very important for the understanding the disease also.
Okay, yeah. One question here is that, do you show this graphical interrelation between different things? This kind of work of understanding the relationship, is it happened more in the last 20 years, last 30 years? What's the time frame of this knowledge that we have gained?
Last 2 decades.
Last two decades. Very, very new.
Very, very recent. Particularly, it started with the Drosophila work again, I would say, because Drosophila, I would say that I will convince you that Drosophila is really good model. But the most of the genetic tools were developed in the Drosophila. You can pull down now one protein, and you can pull down all the interacting pathways and things like that. So most of the genetic studies started with the Drosophila, but today it has been extensively studied in the other organism also.
Obviously, so, with this, which means that you should be able to sequence the genome. That's also probably very, very important.
Yeah, so the genome can be sequenced within 2.5 days today.
No, I understand, but I'm saying that 20 years back, 30 years back, sequencing the genome probably, human genome sequence was done in the 90s. It took like $1 billion to do that. Right. So that was a big step in a sense, if you think about as a science, to be able to first being able to actually do the sequencing of any species, that allows you to make all the progress that you saw in the last 20 years or not going forward.
I agree, Dave, with you, but the genome sequence was not only the thing what happened in some of the model organism like Drosophila. So the Drosophila and the almost human genome came within few months time. I mean, but it was not only that. What happened was that the ability of people to make transgenic animals so easily, right? For example, today, all the predicted genes, we have way to knock it down in a tissue specific way. For example, in Drosophila, any tissue can be taken and we can knock it down in the tissue specific way, and we can find out through transcriptome what are the genes which are expressed in the every single cell of the Drosophila. That is available data. Then the most important thing is that the physical interaction which I am showing here. It is a combination of both genetic data as well as the proteomic data. And the proteomic data was the most important thing because people were able to make, again, transgenic animal to pull down the interacting partners for the every tissue, every organs, which, would have taken at least years in the other model organism.
I see. Got it. Okay.
Yeah.
I have one question. Can I ask?
Please. Yeah.
What is the similarity of Drosophila model with the human genome? I mean, how similar it is?
So at the genomic level, it is only around, say, 65%. but if you look at all the disease-causing genes which people have identified from human, up to 78% of the disease-causing genes, they have repertoire in Drosophila.
Okay, thank you.
Right. Is that answer your thing? Right. Okay. Yeah. Okay. Right. So that is what I told you, that how we can use this model organism, as you asked very nicely, Das. The idea is that if you have the similarity, we should be able to exploit it. For example, say even the yeast today has been exploited because yeast also has the same metabolism like the human, right? Okay, some of the many things are conserved, so yeast become really easy, and every metabolic pathway which are conserved between human and the yeast, they have been replaced by, for example, human genes in yeast. Okay, because it is easy to transform yeast, for example. Okay, so it is a fantastic work from my colleague Asik Khusru, who works in the Canada. So they have replaced every metabolic genes and put it in the yeast. So at the moment, what we are trying to do is trying to understand the basic things, and every as I told you, every organs, how they develop, what are the genes requirement, that is where we want to know, and what are the disease that results out of the misexpression or the not expression or the overexpression of these genes, so that we can screen for the drugs, or even if we have to do gene therapy, we should know that how much amount of gene we need to put it back and things like that. Okay, so that's the crux, and this is what in the next two slides I will try to why we use a model organism.
Okay.
So choosing a model organism depends on say if you are working on the disease. As I told you, if you are working on the glucose metabolism pathways, I mean, you don't have to work in the very high and evolved animals. For example, say working in the mouse is very expensive, and in India, having a mouse knockout is like 4 lakhs, and how many university will be able to have a single 4 lakhs rupees to just do it, right? So it's very difficult, and so the yeast or any other organism, you can use it. So the etiology, what kind of question you are trying to address, is very, very important, so that whatever you know from the model organism should be able to correlate with the human disease condition that you are interested in, right? So the etiology of the disease phenotype is very important, and many, as I told you, present therapeutic is about the relieving the phenotype, finally the phenotype which you see, and we don't look at actually what time the phenotype should be relieved and things like, just like I gave you the example of the brachycardia. So you look about the genes which are involved in the brachycardia or the say pacemaker only when the person comes to the hospital, right? Okay, so the idea is that can you identify say a individual, at least today we know that at least before 15 years before, for the Alzheimer's disease, for example, say a person needs to just give a cerebral fluid, and we can for sure can identify that whether you have a risk to Alzheimer or not 15 years before, because otherwise when the people yeah, people start if people come to hospital after having the Alzheimer, there is no point. Even if you remove the whole plaque, right? People are able to remove the plaque today. It is not going to restore the whole your memory and things, right? So the Alzheimer's disease,
almost like it is a preventative step.
Yes,
would be so much better if we can get there, right?
Yeah, cardiomyopathy, diabetes, Alzheimer, Parkinsonism, all these things are preventable. I mean, if you take the right kind of advice. I mean, yeah, many a time people will say that, okay, don't eat too much carbohydrate, you are predisposed to diabetes. But when you have the party, you won't mind to have few more drinks, extra
of course,
drinks,
right. You cannot avoid, it's a human nature. So
yes. So the whole point from my personal thing is also that as you talk about preventive care. And the other thing which I'm excited about is bringing about a local health tradition in the whole this disease management. Not only the approach most of the Western scientists do, looking at the only at the phenotype, but I'm interested more also interested in the, you know, before the phenotype exists, at least in the our local health tradition like Ayurveda, just by looking at the Prakriti, people can predict whether you are predisposed to say a specific disease and conditions, and you so that you can be treated beforehand, and you should not suffer from this disease condition.
That's
so
a very interesting point,
yeah.
Upendra, which is that, I mean, how do you know kind of this sort of the parquetry that you talked about? How do you sense it, what to sort of look for, you know, instead of, you know, looking at what you said, the Western way of looking at it? So
yeah, so the Prakriti is something, you know, we feel kind of like metabolic thing which you know will vary, but something which can be sensed at the human, and people have been trying to figure out the Prakriti for the animal, but without much success. But at least what we also know is that the when it comes to gut microbiota Prakriti, which people have identified, at least you can mimic those in the animal, higher animal models, and you can look at the if you can relieve those disease conditions. But today there are many institutes in India who PIs, I mean, in other words, they have been given money and they can identify based on, you know, by using the what I call as the scientific instruments, by the texture of the skin or the texture of the sweating, you know, the way you behave, all these, by looking at the whole thing, what people, if Vedya of Ayurveda could predict it, the Prakriti today, by using scientific instruments, you can predict a person's Prakriti. So it is possible. I mean. So we have now in India Ayur genomics, where we look at the already there are few published papers already which were published in the BMC journal Genomics. So we have look at the SNPs variations amongst the Prakriti individual, different Prakriti individuals, what Pitta Kapha we call it, and even the epigenetic changes which are prevalent in the all these different at least the major Prakriti, what we call as the Varpita Kapha. But you know, we including we are looking at it because I'm also a adjunct faculty to what we call as a transdisciplinary university, which look at the local health traditions. So we look at the gut microbiota correlation with the Prakriti.
okay.
Okay.
Thank you.
Is that all right? Yeah. So as you said, model organism, it should be a non-human thing, but it is a in vivo model, because unlike the cell culture system where, you know, you have a 2D, people are claiming to be the 3D. Today we know that you know, there are extensive crosstalk between the organs, and so this should allow to study in the model organisms. Okay, so that is the whole point of using the model organism. And as we talk about, the model organism is something which is non-human, and whatever we know from the model organism, if it is a disease condition, it should have a relevance to the human condition or the disease that you are studying. That is where the precaution is, that the many people in the world overclaim things, and I don't have to exert it from the studies done over the years in the, say, published in the Cell, Nature, or Science. At the Merck company, we have looked at the 69 drugs which people say they can cure cancer. Only 5 were reproducible. So I mean, you know, we are talking about such research. I mean, so the model organism, cell culture, there is a huge difference. So it comes to model organism. Being in India, it is all about you know, cost and accessibility. I mean, you know, because if it is not accessible, if I want to work with a mouse, it is not accessible. If I can't maintain them because it is an expensive thing, there is no point, right? So it should be cost and accessible, as I said. And the gene, whatever you are interested in, whatever I'm interested in the disease, that gene should be functionally conserved, not only functionally conserved in the sequence wise, everything we think, and we should be able to generate knockout, knock-in, or any kind of site-directed mutagenesis using the CRISPR-Cas9 and all those things. It should be easily available, and that is where we work extensively with the Drosophila, because there is no other organism where the genetic tools are easily available as much as in the Drosophila, right? The other thing about the Drosophila is that all the people working in the world, I mean, we don't care about the personal greed that, oh, this I need to publish first, then only I will share with you. No, nothing like that.
I see. Very good.
So the whatever we develop transgenic animal or say the proteins or the anything, we just share.
That's very good. It's a
So the
benefit of science and eventually drug discovery. But you made a very important point. I want to go back to that, which is that the cost of doing this sort of research is so expensive that you have to take that into account in terms of trying to decide which direction or what sort of organism you want to actually study. I think that's a very important point for budding researchers to know. This is always plays a role. I mean, we think that research is research, anybody can do it, but cost always plays a very important role in there. So I just want to re-emphasize that. So yeah.
And the good thing about India is that at least when it comes to Drosophila, we have a huge Drosophila community, the biggest, I would say, other than the, of course, the bacteria or the cell culture people. If you are looking at the model organism, we have, as of this month, we have 179 PIs actually working with the Drosophila in India, and I'm one of the Drosophila board member in India. And you need to just request. We have Drosophila stock centers in India, and we have a community, Drosophila community, where you can just put what Drosophila line you need it. And if you can't get it from outside international Drosophila stock centers like Bloomington or the Vienna or the Japan, we can get it when we are ordering it, and we can supply you. I mean,
that's very good.
So
that's very good.
We help each other a lot in India, particularly for the Drosophila people. I mean, so that's the good thing. So anybody can just look at the Drosophila in Drosophila, what do you call as the fly lines available with everybody, which is attached to ICER Pune, and you can order it, right? Okay. So the disease condition, what we want to recapitulate as much as possible. That is why we work, for example, whatever said and done, we work extensively with the muscles, because muscle is one of the highly conserved tissue amongst all the organisms. Okay, whatever said and done, muscle, the same protein do the same thing. For example, worms to human, the muscle, we work extensively with the muscles because the same proteins are organized in the same way. That is why we work. Up to 96% of the human protein, they have autolog in Drosophila, except for the intermediate protein, intermediate filament proteins, and they do the same thing, right? The other thing is that like tissues like neurons or the whatever disease you want to generate, neurodegenerations or the things, we were also one of the group who generated the human apolipoprotein mutants, the humanized Drosophila or the humanized zebrafish, and we screen for the many say plant extracts and the plant which you can just chew as a pickle or things, and which can remove, which act as a neuroprotective. Okay, I'll come back to that. So these are some of the classical model organisms which people have used extensively, like say yeast in amongst the higher in eukaryotes, Drosophila, C. elegans, zebrafish, mouse, or in plant Arabidopsis. But in our lab, we started working with the Drosophila because I was not only trained in Drosophila, but Drosophila genetics. As I said, if you put all the data from all the organism in one side of a weight machine and you put the Drosophila literature on the other, Drosophila data will be still heavier. I mean, that much data. We are talking about stem cells and everything today, but this whole pathway were dissected in the Drosophila stem cell in 1980s, for example, right? Okay. We are talking about the heart development, Tin Man and all those things were dissected at least in 1992 to 95 in Drosophila. So up to some extent, Drosophila genetics, I mean, we are much ahead of the other model organism. As I said, we have now RNAi for the every predicted genes for the Drosophila. I mean,
so
all
I say very interesting. So, sorry, go ahead. I have two questions.
So all the RNAi that you want to use for the knockdown for the any even predicted gene in Drosophila, I mean, 14,000, but we have close to 44,000 such RNAi because you want to have two for the each one, right? So 3 are there, 4 are there. So we targeting the same gene from the different regions. So the off target should be limited. So we have this RNAi lines which was made at the Vienna in Austria, and some of them were made in the Howard Hughes in America. And these all are available with a nominal price. So if you are ordering from Bloomington stock, I mean, we have to spend like say $20 per strain, which compared to Jackson's laboratory mouse, as I told you, 4 lakh rupees. I mean, it's much cheaper. I mean, right? Okay.
So I think you sort of answered the question, because I was trying to understand when you said Drosophila data, you sort of addressed it that say you have lots of Drosophila data. So just for the audience to understand, what do you mean by the data? It seems like it's related to RNA, and depending on which direction you kind of look into it to get that information.
Yeah. So
right?
expression, tissue specific expression of any gene is known. It is there in the what we call as the fly base. So the fly base has, you need to just put it www.flybase.org. So then if you want to look at the every organ expression level, then there is something called as the fly atlas. So then you have different genome center. I mean, to look at these single cell transcriptome, for example. So all these are there. I mean, so and all the genes are cloned. So you can get it, for example, in India, you can get it from Professor Giris in the ISA Pune. You can just order it, and all the mutations. Other than the RNAi, we also have the knockout line for the 80% of all the genes. So anybody can order it. And
okay, very good. That's good to know.
And the best part is, as I said, I mean, you are just interested in the neurons. And if you want to make humanized Drosophila, I mean, putting the human sequence into Drosophila conserved sequence, like us personally, we did the apolipoprotein E3 versus the E4 humanized one, we generated it, and we could drive it only to the neurons, and we have shown that the E4 is the one which can cause more ROS, right? All these things can be generated in India if you don't have the facility, but if you have a money from your project, you need to just pay around 30,000 to generate a transgenic. I mean, you can outsource it. So we have a outsource place in NCBS in Bangalore. CCAM, we call it, and anybody can just provide the gene name and the details, and they will generate it for you.
Very good.
So Deep, that is why we work extensively with the Drosophila, because today, as I said, any animal, as you said, any animal can be used, but the cost factor is the major thing particularly when you are working in India. I mean, so we do, like places like in the IAC, we do have mouse models. I mean, we have facility for up to say 5,000 mouse different strains, but we can't generate like all the 23,000 genes mutations I mean, there is no such facility in at least in India, but we can generate such thing in both Drosophila and zebrafish. You don't need very big room. I mean, all the we have up to 3,000, and this 3,000 we keep it in a small room with the AC. That's it. You know, so you don't need a big place. And of course, for the zebrafish, you need slightly bigger than the Drosophila, but and the water system, circulatory system, and things like that. But we use zebrafish, but because whatever we can't address, like say eye development, is zebrafish is closer to the human. That is why we use it. When you asked about one of the questions, like how you choose it. So it is a trial and error, because like say mouse is closer to human, but people have generated mouse knockout for the protein called the dystrophy, right? I told you about the muscular dystrophy, which causes the muscular mouse, when you knock it out, it doesn't give the Dussen's phenotype. I mean, dystrophin phenotype. I mean, it just give the Becker milder version, Becker phenotype, right? Okay. Whereas the same gene, when we now knock out in the zebrafish, sub-G mutant, we call it, we see the same phenotype. So if it is a disease relevant, then the zebrafish is more relevant. But a mouse doesn't show phenotype doesn't mean that it is not useful, because from the mouse data, at least we know that there is another protein called the eutrophin, which is compensating the loss of dystrophin. And the dystrophin is also a spectrin binding protein. And we know today there are mini version of the dystrophin, which is like more of like the eutrophin, and you overexpress it. The idea is that it should be able to rescue the phenotype. And so every model is useful, but I mean, at the what level you are looking at the, to mimic the disease condition and rescue it. There is not much time, but I will just run through some data. So we use
so before you go to the data, maybe I ask you a general question here. I mean, with all of these things now, the collecting the data and whatnot, the computing technology also plays a very important role in your research, I presume, because you have to compare information from different ways of collecting the data. If you're planning to talk about that towards the end, that's fine. I just wanted to remind you about something to talk about that part. So. Hello? Your audio is not coming. Let's wait for him to get back on. Upendra, I can see you on the participant, but your audio is not connected. So hopefully it can get connected. Your slides are coming back on, but I cannot hear you.
What is my video now?
Okay.
Professor Deep, can you hear me?
Yeah, now I can hear you. No problem. Okay.
I mean, I'm sorry about the whole thing. I mean, right?
Yeah. No, don't worry. That's okay. I mean, technology is not perfect, but it's much better than what we used to have. So, okay. So
right.
you are going to talk about something towards the end, I think. So please go ahead. So.
I mean, I'll just show few. I'll just browse through the data. I mean, so that you have at least the flavor of what we work on. So
okay.
we put a lot of this human similar protein back into Drosophila. The reason I have told you that particularly we work with the Drosophila muscle protein because they are highly conserved and they do the same kind of phenotype. And these are the Drosophila human disease condition which are being looked at the in using Drosophila world over, okay, including some of us. And we work with the Drosophila muscles because you can see, for example, say here there is no difference in the last corner. So there is no difference between the human and the Drosophila. But the good thing about the Drosophila is that we can trace from the myoblast level to the full structure. I mean, every stage of development, we can trace it so that we can find out where the disease arrived. And so we look at the what we call as the nemaline myopathy, which is caused because of the mutation in the actin protein, which is the one of the thin filament protein. And we put the same, these are the human phenotype, you can see it. We put the same thing in Drosophila and they show the same kind of, similar kind of phenotype. And the good thing about this knowing in the Drosophila is that, as I told you, we can test now that whether gene therapy will be possible or not. We presume that every mutation gene therapy will be possible, but it is not so. Because there are mutations called as the say antimorphic. Antimorphic are nothing but the like a antisocial individual. So the one individual can poison the other thing. In the same way, there is a antimorphic mutation. Then that mutation can poison even the wild type copies available. I mean the proteins available. So such mutations are there. Then the gene therapy may not be functional. So
I see. Very interesting.
The null, if the protein is not there, up to some extent, you can see the last one, 1888F, you can rescue it. But the antimorphs, you can't rescue it, even if you do the gene therapy. So this kind of things, we can predict it by using Drosophila. And the Drosophila, as I told you, transgenic can be met very nicely within 3 months. And so counseling become for the doctors, helping the doctors become easy, because many a time doctors will not know. For them, say mutation in a gene is same for the every all the mutation. But what we can tell is that no, even in the nemaline mutations, individual can suffer a different way. I mean, so this is what we do, and we do the same thing with the what we call as the hypercontraction, but this is similar to the cardiomyopathy. So the more force produced or the less force produced, we can look at it and we can look for the how to make it functional. Just like I told you, the pacemaker, you can increase the heart valve, contraction in the similar way. We can make the Drosophila which are not flying by modifying the things. And all this genetic study, we do it by just putting lots of
human mutation back in Drosophila, which is possible. I mean, I'm not going to get into the whole thing. So even the protein say gene interaction, somebody will have cardiomyopathy, but another may not have it. So the such patient related things we can study it, because one may have what we call as the modifier in the another genes, and which may not be present in the other individuals. So where there is a modifier which will enhance the phenotype will suffer from cardiomyopathy, but whereas where there is a modifier which is what we call as a suppressor, then the individual even after having the genotype need not suffer from cardiomyopathy. All these things can be tested. The most important thing is that once you have all these phenotype, then we can develop new technology, and this is what exactly what excites me. At least here we develop say calcium sensor in Drosophila, which can at least tell 60 hours before the phenotype, the actual muscle pulling starts. Calcium abnormality, you can see it. We are talking about in the human condition at least 20 days before somebody suffer from the heart attack. If you go to hospital or for the routine tests, and we just need is a one cell from the cardiomyocytes, right, which you can get it from your blood circulation, and just by looking at the calcium imaging thing, we will be able to tell that no, you may suffer from the heart attack, you should take the precaution. Okay. So the preventive kind of thing we can do it, and this is what we developed. So we put many of these human mutation back in Drosophila so that we can understand the etiology, but also look for the drugs which will recover it. So for example, say we met the Alzheimer's disease condition in Drosophila, then we look for the neuroprotective plants, like say in Karnataka, they use one plant, small shrub called as Decalapis hamiltoni, and the people, those who consume this plant, today we know that they don't suffer from neurodegenerative diseases. I mean, it acts as a protective thing, right? And we here what we have shown is that how the compound present in the plant can be useful, and how it is doing, that is what the mechanism we have shown it. And the same way we are doing with the epilepsy, because at least in India, 16 up to 9 to 16 individuals suffer from the epilepsy, and we identify from the Ayurvedic literature that giving pomegranate helps in elevating the phenotype of epilepsy. And today I'm happy to share that, because we are close to publication, so I'm happy to share that we have identified the compound which does that. I mean, and
that's
I don't
good.
want to I don't want to show you the data, but the epilepsy model, you can see it, how the fly behave, just like the what we call as the uncontrolled movement, right? Yeah. So you can see it. I hope you can see the movies. I mean, right? So this is how we generate the epilepsy, and we look for the compounds, and so we generate today. We are able to identify the urolithin a compound is a aromatic compound, can actually suppress the epilepsy conditions, and excited about it, because we are the first to talk about that urolithin can suppress the epileptic conditions. I mean,
this is fantastic.
Let's not get into it. At the same way, as I told you.
Let me stop you for a second. Somebody has a question. Panchalika, you have a question. Go ahead, please.
Yeah.
Unmute yourself.
Hello.
Yeah, she has raised the hands. Panchalika, did you want to ask a question? Okay. I cannot hear you. So go ahead, Upendra. Continue.
Yeah. Similarly, then what people say is that Drosophila is the insect, and the most of the bigger journals will say that no, you need to work with the vertebrate model and things, and that is right, because there are similar kind of gene setup. So we have only one copy in Drosophila, but there are multiple copy of a gene in vertebrates, including human. So there could be redundancy, means that the, one gene mutation, it could be there, then the other may take it over the function, right? So the such compensatory things you will not understand working in Drosophila because you have only one copy. That is where we work with zebrafish depending on the human again disorder. For example, say the Drosophila eye is compound eye, but if we want to look at the eye, then we look at the zebrafish eye, because the zebrafish eye development is equivalent to almost like with the human. So we use extensively for the drug screening, knock down again CRISPR-Cas or the Morpholino. All these techniques can be used to knock down any gene at any level in zebrafish. And what here I'm just giving you example our recent work where we have put the human mutation for the first time in the world. We were able to show that the mutation in a protein called as the WDR8 causes microspherophakia, or other words, small lens condition, which we identify in two family in Bangalore. And we were able to show that it is because of mutation in a protein called as the microspherophakia. And as I told you, how you will know that this disease is because of this gene, right? The best part is that when you can show by rescuing the disease condition with the wild type phenotype or with the mutant conditions, right? And that is what exactly you can do in zebrafish or Drosophila easily. And we were able to show that wild type humanized zebrafish, we call it, wild type can rescue the phenotype, but the mutant which we have identified can't rescue the phenotype. And once we have this phenotype in the model organisms, remember that we can't kill the human, but the zebrafish can be killed up to some extent with the ethically can be killed, and we can look for the mechanism. And the mechanism we were able to show that how the cell cycle progression are affected. That is why you have the small lens in the zebrafish or in the human conditions. And once you understand that this mechanism, then it will become at least easy to say that how we can stop it. That's the crunch of the why we are excited about all this model organism, right? Okay. So we do the same thing with the actin nemaline myopathy in the whatever we did in Drosophila. We do that in the zebrafish also, and we look with that. You can see different amino acid which doctors are giving in the these patients. So we have generated these conditions, and now we can test that which doctors are giving to the patients will be useful. And you can see the data that not much of this whatever the food supplement people are giving is not that useful. I mean, okay. So that's the conclusion we can do it. And so the take home message is that sorry that I have taken longer time, but the whole point is that the model organisms ultimately what we want to generate is that even for the normal condition we want to generate, because there is nothing like normal is in the Gaussian distribution. You have the median, which is the normal supposed to be, but you don't know, depending on the which phenotype you are looking at, you could be suffering from any defect, metabolic or the protein or the gene. So we want to generate model organism so that at least we can look at your avatar, including your how long you will survive if you take, say, lots of protein, if you take lots of carbohydrates, if you have a predisposed with the diabetes, what kind of food you should take it so that you can maintain your body weight and things like that. That's the crunch of the whole thing. And what we are as the individual looking at is that how we can manage and prevent disease condition before it really happened, or before it really cripples our daily work. I mean, that's the take home message. Right. I stop here and I will be willing to take any questions if anybody has.
This is very fantastic. And I can see the excitement of what you do and what you are doing. It just makes me feel like maybe I should go study under you and with your excitement of doing new stuff and everything. So I'm going to ask one question and then I'll open it to the participant. You can unmute yourself and ask questions instead of typing. The first question is that what made you, I mean, it's more a understanding, as a researcher, we progress through, different stages in our research. And what is, from my understanding, your PhD dissertation work or postdoc work was not on Drosophila much at all, but you kind of moved into that knowing that there is that there's something new out there with the, sequencing and whatnot. So is there a particular moment in your life or in your research life you felt like that, this is really where I want to kind of go to, and this is, can you share anything in that direction?
So Deep, I actually worked with Drosophila throughout my career.
Okay, you did. Okay.
So not with the zebrafish.
Not the geographies. Okay.
So Drosophila, when I completed my master's in 1994, I'm not trying to boss it, but I was a student politician, involved in the kind of like secretary of the student bodies and things. And my father actually wanted me to become go for the civil service in India, particularly from Northeast. The thing is that if you are a gold medalist and things, you should go into civil service, and so for me, the exciting thing was that in 1995, 3 people from Drosophila, I was interested in Drosophila because the Mysore University is known for Drosophila work. But what excited most was that in 1995, when 3 people, Edward Lewis for the discovery of the Hawks gene, like 4-winged fly, and Eric Weishaupt and Nusrin Walhart. So these 3 people got the Nobel Prize in Physiology. When I completed my master, I said that nothing going back, I should work with Drosophila. And so, and then I wanted to work on something which not many people are working, and at least in the muscle field, we had 2 schools. So the one, those who are looking at the patterning of the muscles, and the one, those who are biochemists and protein people, those who really look at only at the muscle protein and things. And I said that the patterning artifact, of course, then the muscle structural protein also, there should be some crosstalk. And there was no one at that time working on this interface.
Okay.
So you have developmental biologists and you have biochemists. And they won't talk to each other.
Of course not. They won't.
So being a geneticist, I wanted, to fill up the gap, and that is where I thought that I have a niche, and of course, I started working on things. But as soon as I joined IAC, I always wanted to come back to India because I had an offer from outside after completing my postdoc. But I don't know what you said. I was too patriotic. I'm not telling that those who are working like yourself, you are not patriotic enough. But I was always attached to like India, or say, if I'm in India, I thought that I will be able to help more of like a Manipur or the Northeast region. I will be able to help. And other thing which made me come back in IISc was that there is an incident when I completed my 12 in Delhi. When I came back to Bangalore for the first time, the first PhD scholar from IAC, his name is Lalit Pukhamram, our first Manipuri. He was going for US. He's a professor at the Michigan University, and he works on diabetes. So he was living for US as a postdoc. And I asked that, what is that you have done? But then I didn't know what is the expansion of PhD, okay, by the way. And he said that, just 12 standard. He said that I'm the first one from Manipur, and I said that this institute look fantastic. So you have been to IAC. If you come through that road, no, it is green.
Yeah, I have been there.
And I asked to Tamo Lalit that is there anyone from Northeast? In how many of you are there? Then he said that there is no faculty. And I don't know. I have this what you call deja vu kind of thing, that I wanted to be the first. And then I went back and look at what is the expansion of PhD, what to do, and things like that. And I would say that I am not a very intelligent person, but
I,
I'm a
I
hard
I
work.
would disagree with you on that one.
No, I work
From
hard. I
listening
work
to you
hard.
for 1 hour,
Being
so okay, so
being from the Northeast, I work hard. I mean, so
yeah, of course.
I told to myself that what is the requirement to come back to IISc,
Mm,
and I had that thing that nobody has worked in Drosophila. There was no Drosophila lab in IISc in 100 years. I am the
mm,
only one,
I
right?
see.
For
Very
example,
good.
so
Yeah.
and nobody worked on the muscles, and
Hmm.
drifting little a bit to neurons also, because neurons and the muscles, they work in tandem together. Um, but this is where I would like to encourage my students and a colleague from the Nordics. I mean, you dream big, and people say that you get disappointed if you dream big. I mean, for me personally, I dream big. I mean, people used to laugh at me. I mean, I never hesitated to tell people that I want to be in IAC. And those who have seen me in BSc and masters, they used to laugh at me and say that a person who can't, because I study in the Hindi medium in Delhi,
Hmm.
people used to say, you can't even speak a English sentence properly and you want to be in IAC. God,
Yeah.
and I do believe in the Paulo Coelho in that way, that
Yeah.
you should want something desperately, the whole universe conspires that you get it, and that is my thing. I mean, so I'm here, and the zebrafish, because over time I realized that we need the zebrafish work to complement the work that we are doing in Drosophila. I mean, that's all. I mean, when it comes to human condition, so. We are the only group in IAC still working extensively on Drosophila and zebrafish.
Fantastic. This is very, very heartening and good to hear. Let's see, does anybody have any question from the participant? Any general question or specific questions? You can unmute yourself and just start speaking.
I think we were the only people talking.
That's why I said it's a fireside chat, sitting in Imphal, by the fire on a wintry night, sort of an idea.
Yeah.
That's what the whole idea is. Still, I'll give one more chance. Anybody have any questions?
And by the way, Deep, this is for the people, those who are 15, 16 people. People think that being in IAC, I could be reserved, not approachable. I'm not. If anybody
Yeah,
has
right.
any kind of interest in the science, and if you want to look at, and some of you are senior there, and if you want to collaborate, I'm open to collaboration, because I extensively collaborate with people all over the world. If you look at, I work from, by the way, it is not necessary that I should work only, it should be only Drosophila. I have helped people in the Northeast to work on bacteria. If you look at my publication list, you will see that I have published in bacteria identification of a bacteria which has got ecological translational potential. Recently, we met a transgenic silkworm with putting innate immune gene from Drosophila back to silkworm, so that the silkworm can confer more resistant to multiple pathogens. I mean, because the silkworm didn't have these antimicrobial peptides. So I am always looking for some translational work. I mean, so we were the first to make such a silkworm indigenously from India. And if you look at, we just made it last week, the paper was published in the molecular biotechnology. So we are happy about it. So I'm open to any kind of collaboration, provided you have a strong question to address. A strong question means that scientifically novelty is very, very important. And
very
so
important.
something,
Correct.
yeah,
Right.
right. Okay.
I think Upendra, you made a very important point. I want to reemphasize that. Many people, especially even students, they feel like that we are not approachable because we have been in the field for whatever 20 years, 30 years. That's an impression I had when I was a student, that I cannot approach somebody who has been there. But what I have found over the years is that there are obviously exceptions, but most people in our scientific field are approachable. You can approach people to be able to ask questions. Sometimes they do not respond right away. That's not because they are trying to ignore you. somebody like me, I get 200 messages per day, sometimes email messages. It's just impossible for me to remember who actually sent, because I don't know your name yet. So if I have to think about what was the name of the person who sent me an email 2 weeks ago, I would not be able to recollect that. So I used to have a message on my webpage saying that if I don't respond to you in 2 weeks, please contact me again, because that's it. So I think that it's very heartening to know that you have the same sort of a philosophy, that approachability for other people to, want to do.
Yeah, I mean, so IAC professor used to be like that. I mean, our seniors, I mean, at least few decades back, they were not so approachable. And
no,
they used
that's
to
a
think
different
that,
time.
Yeah,
That's a different time. Yeah, right.
Now we are different. We are different.
We are very
right?
different.
Yeah.
And science is very exciting also now. There's so many things you can do with it. So, all right. I don't see any other questions. So, Upendra, I really want to thank you so much for your enlightening
it was a
talk.
pleasure. It was a pleasure.
Yeah. So with that, I'm going to stop it here. Okay.
Right. Thank you very much, Professor Dept again.
Okay.
Yeah. Thank you very much. Yeah.
Thank you.
Yeah. Anybody, if they have any questions, they can always write back to me. My official email is upendra@iiac.ac.in. I mean, anybody can reach back to me.
Okay. Sure.
Yeah.
Thank you very
much. Yeah.
Okay.
Thank you very much.
Okay.
Yeah.
Bye-bye.